All right, today we're going to dive into some pretty amazing technology that, believe it or not, lets us listen in on the tiny conversations happening inside our very own cells.
I mean, just think about that for a second. What if you could take a snapshot right now that shows a unique pattern of all your active genes? A pattern that basically defines what your cells are up to at this exact moment. Well, that's not sci-fi, it's the real power of gene expression analysis.
So, why is this such a big deal? Why does it matter so much? Well, it all comes down to a really fundamental idea in biology: gene expression.
The easiest way to think about it is like this. Your DNA is this massive library of cookbooks. Gene expression is the act of actually picking out a specific recipe, that's the gene, and cooking the dish, which is the protein. So when we study this, we're not just looking at the table of contents. We're seeing what's actually being cooked in the kitchen right now.
And this isn't about looking at just one recipe at a time. No way. The whole point is to get a global picture, a huge, sweeping overview of thousands of genes all at once. And that gives us this incredibly rich understanding of what a cell is doing, how it's behaving, and maybe even what it's about to become.
So, the takeaway here is that this snapshot is unbelievably useful. It helps scientists get a handle on everything from how a single cell can grow into a complex organ to what's going wrong in diseases like cancer. It lets them put a healthy cell right next to a diseased one and see exactly what the genetic differences are. But how do you actually take this genetic snapshot? For a long time, the go-to tool for this was a really remarkable piece of technology.
Meet the DNA Microarray. It's this tiny little glass slide, usually no bigger than a postage stamp, and it's covered with thousands of microscopic DNA spots. You can think of each spot as a tiny probe just sitting there waiting to see if its specific gene is active in the sample you're testing.
Probably the best way to visualize this whole thing is to picture a massive, old-school telephone switchboard. When a gene is active, when it's on, its little spot on the chip lights up. If it's inactive, the spot stays dark. So by looking at this whole pattern of lights, we get our snapshot of what the entire genome is doing. Now, getting this pattern of lights is one thing, but actually making sense of it? That's a whole other story. The raw data that comes off a microarray isn't clean at all. You have to find the real signal that's hidden in all the noise.
What you get is just a flood of numbers, and it's full of all these little technical glitches and background static. The real magic, the real science, is in the data analysis, the process of cleaning up this messy data to uncover the true biological story buried underneath.
You know, a really fantastic analogy for this is digital photo editing, right? You take a raw photo with your camera, and you have to adjust the brightness, fix the colors, maybe remove some dust spots, all to make the real image pop. Well, prepping microarray data is almost exactly like that.
This photo editing for genes basically follows a three-step process. First, you do a background correction to get rid of all that random noise. Second, you normalize the data. That's like adjusting the brightness and contrast so you can compare different photos fairly. And finally, you summarize it all to create one clear final value for each gene.
Okay, so let's dig into that a little bit. Let's look at each of those photo editing steps just a bit more closely.
First up is background correction. This is all about cleaning up the initial image. You know, the scanner itself might create some optical noise, or some random molecules might stick to the chip where they're not supposed to. This step is all about digitally subtracting that background hum so we can actually hear the real signal from the genes.
Next up is normalization. This is so crucial. Imagine you took two pictures of the same person, but one was in bright sunlight and the other was indoors in a dim room. You couldn't just compare them side by side, right? Normalization basically adjusts for those kinds of technical differences. It puts all the data on the same scale, so you can be sure that any differences you see are actually biological, not just some fluke from the experiment.
And finally, there's summarization. See, on these microarray chips, a single gene isn't just measured by one spot. For accuracy, it's usually measured by a whole set of them, sometimes 11 or even more. Summarization is just the process of combining the data from all those little spots into one single solid measurement for that one gene. It's how we get our final confident number.
But here's where it gets even trickier. Some of these chips actually include special probes called mismatch probes, or probes, and their entire job is to measure that background noise we talked about. The problem? Well, sometimes these probes are themselves pretty unreliable, and they can end up introducing more noise into the whole thing.
And this really shows you that data analysis is part science, but it's also part philosophy. Different algorithms take totally different approaches. So one algorithm called MAS5, it tries to use that mismatch data and correct for its flaws. But another really popular one, RMA, it just throws it out completely, basically arguing it's just too unreliable to be useful. There's no single perfect answer, which is why choosing the right method is so important.
Okay, so now that we get the process, let's zoom back out. Because like any really powerful technology, microarrays have some absolutely incredible strengths. But they also have some important limitations we need to talk about.
I mean, their power is just undeniable. They give you that global view, analyzing thousands of genes in one go, something that was just impossible with the older, slower methods. They're also designed to be super specific and give you reproducible results, which makes them a really solid tool for researchers.
But, and this is a big but, it is so important to remember the pitfalls. The biggest one is that it's just a snapshot. A cell's activity is changing constantly, every second, and a microarray only captures one single frozen moment in time. Plus, just because a gene is more active, it doesn't always mean you get more of the functional protein that actually does the work in the cell. That link isn't always one-to-one.
And this quote really gets to the heart of it. Microarray experiments, they don't give you the final answer. More often than not, they generate these fascinating new ideas and raise even more questions, basically pointing scientists toward the next experiment they need to run. It's really a tool for discovery, not the final destination.
So we're left with this powerful but ultimately static picture of our genes in action. The snapshot is incredibly detailed for sure, but the ultimate challenge for science is still out there. How do we take all these individual frames and stitch them together to see the full, dynamic, and frankly, breathtaking motion picture of life itself?